Have you ever just stopped and thought about how the human brain actually builds itself? I mean, it is without a doubt the most complex thing we know of, and it manages to construct trillions of perfect connections from what starts as, well, basically a blank slate. Today, we're going to pull back the curtain on that process. We're going to look at the actual blueprint that neurons use to navigate the developing brain and form the circuits that literally make us who we are. So that brings us to the central question for today. You have billions upon billions of neurons. Each one of them needs to make, in some cases, thousands of specific connections. How on earth does that get done with such incredible precision? This is not random, not by a long shot. There is an incredibly elegant multi-stage system at play, and the stakes couldn't be higher, because when this wiring goes wrong, it can lead to some really profound neurodevelopmental disorders. So understanding this blueprint is absolutely key. All right, so here's our game plan. First, we'll get a handle on the sheer scale of this wiring problem. Then, we're going to meet our hero, the intrepid little navigator called the growth cone, and see how it uses molecular maps to find its way. Then things get interesting. We'll uncover a surprising trial and error phase where the brain just makes way too many connections on purpose. And finally, we'll see how competition and activity sculpt those circuits into their final perfect form. Okay, first things first. Let's really appreciate the challenge here. For your nervous system to do anything at all, these long fibers from neurons called axons have to travel, and I mean really travel, sometimes across huge distances, through this dense, crowded, developing landscape to find their exact targets. What you're looking at here are the breathtakingly beautiful drawings of Santiago Ramon y Cajal, who's really the father of modern neuroscience. He was the first person to actually see this neuronal forest, as he called it. So the fundamental job for every single one of those neurons you see is to grow its axon and plug it into the right spot in this incredibly dense network. So how does an axon find its way? I mean, it doesn't have eyes. Well, it relies on this amazing little structure at its very tip. It's a microscopic explorer that's kind of like its eyes, its nose, and its engine all rolled into one. This is the growth cone. But before that journey can even start, the neuron itself has to grow up a bit. As you can see here, it starts out as a simple little cell and then starts extending these small feelers, or neurites. One of those is going to get the signal to become the axon, that's the long-distance cable, and the others will usually become the dendrites, the branches that receive signals. So what is a growth cone, really? It's this incredibly dynamic, moving structure right at the leading edge of that growing axon. The best way to think about it is as a sensory probe that is constantly feeling and tasting its environment to decide, okay, do I go left, right, or straight ahead? And this is just a fantastic close-up look. The growth cone extends these superfine finger-like projections called filopodia. They are always moving, literally feeling their way through the developing brain by sensing chemical signals that basically tell them which way to go. And how does it make those choices? It all comes down to a simple push and pull system of chemicals. You have chemoattractants, which are molecules that scream, "Come over here, this way," and they pull the growth cone toward them. And then you have chemorepellents, which do the exact opposite, pushing the axon away. It's basically a sophisticated, high-stakes game of molecular hot and cold. Okay, so let's zoom out. How do these simple, local push and pull signals create the incredibly organized maps we see in the brain? How do millions of axons use these cues to build a large-scale, orderly system? Well, they do it by forming what we call a topographic map. Now, the idea here is actually pretty simple, but it's so powerful. The spatial arrangement of neurons in one place is perfectly preserved in how they connect to another place. It's like taking a map of one city and laying it perfectly over another, so all the neighborhoods stay in the same relative position. And this is a brilliant classic example, the wiring of our visual system. Neurons in the retina at the back of your eye have to connect to a part of the brain called the tectum. And they create this perfect map by following molecular gradients. For instance, axons that have a lot of a certain receptor are repelled by areas in the target that have a lot of the signal, forcing them to find a spot with less of it. This creates a sort of molecular coordinate system, like a GPS. You know, this whole idea was actually proposed by Roger Sperry, who won a Nobel Prize for it. He called it the chemo-affinity hypothesis. And what's amazing is that he theorized that neurons must have these unique chemical tags or labels to find each other, long before we ever had the technology to actually see the molecules involved. And you know what? He was absolutely right.
Now, here's a really critical point. A single gradient just isn't enough to get the job done. I mean, think about it. If you only had one repellent chemical, all the axons would just get pushed to the other side and pile up where the signal is weakest. To create a smooth, complete map that covers the whole area, you need these counterbalancing forces, a mix of attraction and repulsion, to make sure the axons spread out nice and evenly. Now, you might think that with this incredibly sophisticated guidance system, the initial wiring would be pretty much perfect. But it's not. It's actually more like a rough draft. And to refine that draft, the brain uses this fascinating strategy. First, it creates a massive surplus of connections. This is a principle we call exuberant connectivity. Early in development, the brain essentially hedges its bets. It goes overboard, forming a huge surplus of synapses, way more than it actually needs. It's like a trial and error strategy on a massive scale, just to make sure every target cell gets contacted by lots of potential partners. And the scale of this is just mind-boggling. I mean, read that quote. By the time development is done, more than half of all the synapses that were initially made are gone, eliminated. This massive cleanup job is called synaptic pruning, and it's absolutely essential for creating an efficient, fine-tuned nervous system. So this brings us to the final act, and maybe the most important part of the whole story. This is where that rough map gets sculpted into its final, precise form. So how does the brain decide, out of these trillions of extra connections, which ones do you keep and which ones get the axe? Well, it all comes down to competition. The whole refinement process is driven by two main things. The first is neural activity, basically which circuits are actually being used. And the second is this fierce competition between axons for a limited supply of, well, life support molecules called neurotrophic factors. You can think of these like a special fertilizer for synapses, and the target cells only release a little bit. The connections that are the best at getting this fertilizer survive. The others, they weather away. Let's just break that process down. Step 1. As we saw, the brain overproduces connections, creating that messy rough draft. Step 2. As these new circuits start to fire, the synapses that are part of an active working pathway get stronger. Step 3. Those stronger connections now have an edge. They're better at grabbing those limited survival factors from the target cell. And that leads to Step 4. The synapses that are quiet or just less efficient lose the competition, they get weaker, and eventually they're eliminated. It is a stunning example of Darwinian survival of the fittest selection happening at the level of a single synapse. So let's put it all together. The big picture, initial layout of the brain's maps, that's laid down by those gradients of molecular guidance cues, the molecular GPS. But the final, high-definition, pixel-perfect version of those maps, that's sculpted by activity and competition. It's a brilliant two-step process that combines a genetic blueprint with real-world functional validation. And this whole story leaves us with a really profound final thought. This process of pruning and refining, it doesn't stop at birth. It keeps going, driven by our experiences. So if our neural circuits are literally selected and stabilized based on which ones we use, think about what that means. How do our earliest experiences, the things we see, the sounds we hear, the interactions we have, how do they literally, physically build the architecture of the brain we will carry with us for the rest of our lives?